Hello everyone.
Hello.
Thank you Ashish for sharing a very good informative session on filtration. I hope everybody liked it. To take some good points out of it, I think it was informative, and I see that you were teaching us more on the screening part, like how do we remove all those stuff. So screening, I think it is something also related to differentiating. Do we all agree?
Yes.
Differentiating, do we do this in our day-to-day life?
Yes.
How do we do that? Can we have some examples on that? Can you think on that? Differentiating,
Ingredients while
screening
cooking.
or differentiating?
When we are taking vegetables,
Ingredients while cooking.
No.
By color. We can do it by color.
Yes.
Quality.
Quality. See, I think we have a lot of ladies here. You operate gas, right? I mean burners,
Yeah.
LPG gas.
Even men we also do it.
I mean, I'm not. I'm not being male chauvinist. I'm just saying that generally the usage is with ladies, right? So how do you detect if the gas is leaking?
smell.
Smell, right?
Sometimes it is very mild sound, gas leaking sound.
Yes.
Right, sound or smell. So we are basically able to differentiate or find it. We are able to sense it, right? Can you tell me how much senses we would have it? How many senses? we have? Five.
Actually 6.
6 senses
Yes,
also
okay.
there.
That is debatable. But 5 is something what we have. Do we have like imagine how many sensors are there? Biological sensors are there? I mean, we have 5 sensors.
Sensors. Sensors.
Biological or living organism sensors or senses. Nose, of course, right?
Ears, tongue.
Minus with
But with whom? Only it's with us, or is it with something else? Also?
Some animals. Plants.
Some animals. So
All animals have sensors.
okay,
Plants have.
plants have it. Any example for plant? You remember? Touch me not.
Touch me not. They have
Very
tentacles.
sunflower.
Sunflower.
Picture plant.
So we have plants and animals as sensors, right? Nothing else. No senses to others.
Venus flytrap.
No, there are a lot of technological sensors.
No, of course. I mean, I'm just talking about currently biological, the living sensors.
So you can use a biological cells to make a sensor out of it.
No, I'm saying that biological sensors. Do we have anything more than plants, animals, or human beings?
Insects.
Insects.
God.
God.
You just said oh God. Or
Animals do not have,
microbes?
do not have kinesthetic
Microbes.
I heard of
have
it.
instinct.
Sorry.
Animals do not have what we have. Like they can't have kinesthetic and all. They just have instinct, you know.
I think what Abhay said is very interesting. Microbes. Yes, microbes have it. Actually, if we remember amoeba, the first living organism, it actually eats everything or it does based on the sensors. It has its own chemical sensors. So somewhere our life is always related to sensors. Okay. My topic is something almost related to that or on top of that. So I am going to talk about something on the microbial sensors. I have made a sensor which is oil and natural gas biosensor. So as we discuss about the organisms or the microbes that they have, lot of these microbes they rely on eating something or in some conditions. Sometimes they are existing in hot condition, hot temperature conditions, sometimes in very cold conditions, sometimes in regular living conditions, sometimes even in oil and gas. Lot of bacteria or specific genera of the microbial world, they rely on eating of methane gas and then they live their life. So then I thought that why not I make kind of a sensor which I can use this bacteria and detect somewhere oil and gas. So what did I do then? I did that. I know that okay, I have microbial genera, I can use it, but I need to have some sort of identification that it is detecting. So I realized that I have some sort of identification maybe through light. I think light is something very common we have for identification, right? Do you all know firefly?
Yes.
what does it do, or how does it, what is the characteristic of a firefly?
Light.
Glows in the dark.
Glows in the dark. So it has actually specific genes or DNA, which when it goes into the dark, it would have a certain liquid inside the body that would lighten up, which is called luciferin. And there is a gene for that which is called luciferase gene. So what I did was that I took a DNA and I inserted that gene into that DNA, and that whole DNA or the circular plasmid what I had, I inserted into that bacteria which is eating on methane gas. Now what do I have? I know that if the bacteria is eating onto the gas, it will express that other liquid, luciferin, which will glow. So that means even if I have this bacteria and I throw it somewhere in the dark, I know that if that place is having methane gas, it will eat and it will live and it will give me a light.
So it glows only if it eats the methane
Yes,
gas.
yes. I'll make a DNA which would have it glowing product coming out of its body only when it eats or it lives,
And it is methane.
right? Any queries? Okay. So now, but the only challenge I have is about the signal. How do I know? I mean, imagine if I have an ocean and if I'm throwing these so-called my chips and I'm leaving it there, but how do I know that whether this place is having, I mean, I cannot just keep traveling with a fisherman or taking my boat, right? I would need to have some sort of communication. Now we had very good lectures on communication, we have some very good personalities, we have some electrical engineers or electro communication engineers. Can somebody suggest me the best or easiest way of communication nowadays? Wireless?
Satellite signals.
Satellite signals?
RFID.
Right, I think you are my actually that's what I was going to talk about, and it says there also RFID, radio frequency identification device. It is one of the device which would have radio frequency that I can attach it with my small kit of bacteria, which would give me a signal when it glows the light. So if I am sitting in my center, if I am sitting in my research center, and if I have thrown some of my chips outside in the ocean or anywhere, it would give me a signal. And that is the way maybe I can find it out. So this is what a general biosensor I have made it, and this is the graph that I have it, or the sketch I would have it. If you see my biosensor would be lying somewhere in the ocean. It would have a plate on which the microbes is fixed onto that, and when it eats onto the methane gas, it would release the light. That light would have frequency direct to giving a signal to my center. And then I know that, okay, this is the area or place I need to work on. Now, do we know any general biosensors that we use sometimes in other day-to-day our diagnostics? Trying to be specific on diagnostic, maybe you can think some of the medical persons are here.
Blood group testing.
Blood group testing, diabetes, glucocheck. Right? It has the enzyme that would detect whether the glucose content and how much it is. Based on that, it will have. So basically, it's a biological sensor. The enzyme is a biological sensor. Thyrocheck again. Then there are pregnancy tests, which would have hormone testing kits. So it's again basically a biological sensor. Now I'll try to be, let us do something, an activity kind of thing. You tell me, I have a problem that I have food stuff, which is always like frozen or kept under the cool condition. And can I do any detection whether the really food has been kept under the cool condition or it has been kept under the hot condition and the quality what it has come is not good? Can I suggest some things? Maybe you can take a half to 30 seconds something to just write or think on it. Yes, 30 seconds time. Food nowadays is frozen or under the cold conditions like milk, dairy.
When you get the responses and then you
Yes. Dairies or something, and if it gets hotter, you don't know. But how? Is there any easy detection made for that?
Temperature sensor.
How?
How do you measure temperature?
How do you do that? Of course, you have thermometer, but you cannot just keep thermometer every time in the supermarket, right?
So what do you want?
I'm asking. I am saying that this is an activity if you want to do.
We don't know.
You touch it and you see if it's cold or not.
Yes, I'm very simplistic. Very simplistic.
You can probably the color of the tube is changed.
Keep a biosensor bacteria which will die when it is formed.
Agree. That's a good option. Anything beyond that?
Any indicator which will have
Carry a
color.
biosensor bacteria.
Okay, we don't have to be that much high tech. You are aware of the paraffin or wax, petroleum jelly. The melting temperature is somewhere like 25, 30 degrees, 37 degrees. So anything if it is going beyond that temperature, it's bad. So if we have a Marco, the green or the red, we have Marcos nowadays on the food packets. Imagine if that green would have a wax already in that.
Oh.
You would see that it's molten.
Okay.
Yeah, that's right.
So you
Makes
know
sense?
that it's
Yes,
not
yes.
like warmed and then refrozen.
Right. Even if it is refrozen, it would actually melt down and then it would have that.
Yeah.
The green mark is melting.
Right.
The wax on the green one.
And you have wax with the green mark. Imagine if that is not hot. It would melt.
Then Reliance will not be in business.
Okay.
Thank you. It's very interesting. You made this stuff, is it?
Yes.
It was my research project in my master's.
In your master.
And I want to really make this sensor.
This sounds like incredibly interesting and.
I am sharing something which is not even patented, but I know like
No, we don't have any skill set to take this and do it. By the time we take it, we look at it and forget it. Who's going to patent this?
So you are not.
But tell me, I had a question.
I thought you would Google about it.
Yeah, go with the what?
You don't have patent rights. I can go and share.
No, I mean I would have patent if I make it and then apply it.
Yes, of course. Patent
It's
will
not
be-
only for the
Yeah.
ideas, it's for the execution.
Execute. Tell me something. What is the enzyme you called it then?
Lucifer
Is it because Lucifer is the biblical name of the devil?
Yes.
Luciferin.
So Lucifer.
Luciferin is the protein.
Okay.
Luciferase is the enzyme.
So that's what I was wondering. Has it got something to do with devil or devilish? Because Lucifer is the name for the devil.
I don't know.
666, number of the beast, Lucifer. That's what they're called, right? I thought that maybe, I don't know if the connection is that.
That is actually in the firefly. In the microorganism, it's like methane methoxygenase system. That is again an
I
enzyme.
prefer
which is
Lucifer.
different.
What?
30.
Oh my god. Oh my god.
We should not have
13th participant, Friday the 13th, and he's talking about Lucifer. Alright, so let's have feedback for Parth. What did he do well? What was so good about the facilitation, etc. that he showed?
He kept playing football very nicely.
Playing football as in?
He kept doing with his leg like this.
Acha, okay. So what did we like? So I'm sure you did not like that. What did we like
I
was?
liked it.
Content was amazing.
Yes.
Content was really good.
And very simplistically put. Very simplistically. I can imagine it wasn't that simple when you were researching it in your masters.
Our visualization.
Ideas.
We were actually visualizing everything else, what he was saying. In spite of being so technical, it's not that we didn't want to understand, we were seeking understanding desperately. And
I could see that you
we
got
did.
that and you throwing it into that area where the methane
Correct,
gas is.
yeah.
You know, you did that. So I do and I just throw it there. What would you do?
Yeah.
The glow would come. So you wouldn't tell us what would happen. You would ask us after you told us, so what do you think would happen?
Yeah.
I'd say, well, it would glow. And yeah, exactly. So that's what I'm trying to do. So you're getting us involved and you're getting us to find the answers ourselves. What about this group? What did Parth do well? It's a technical topic, I think, probably the most heavy technical topic till now.
Yeah.
It's the most.
But I never felt that really.
Technical topic.
It was very interestingly
The
put, I think.
passion that he has for the
Yes.
subject came out very
He vouched his interest very well. I
Yes.
mean, it was like, you know.
And
anybody could
I've
buy
written
it.
that exact same thing. Your passion was transmitting to me through the way that you were delivering that. It's like you really believe in it, you enjoy it, you made us have fun in a scientific topic that most of us we've not even started. Probably we've studied what amoebas and all. When have we studied? Ninth standard last time. Last time in 9th standard. And I still mitosis, meiosis. Suddenly I got involved in
Yeah.
it. That's because my mom was a science teacher. She wouldn't let me go. Not that I had any interest in it. Just to let you know.
It was very
What is
easy
the
to
one thing-
visualize whatever you said. No?
Yes.
Everything we could visualize.
Very
You
good.
made the visuals come alive.
I'm glad.
What are the one or two things you would want Parth to do more of as he goes ahead and delivers more trainings in more creative ways. What would you like to see?
You didn't need your slides at all.
True.
Because you were
Because
jumping.
imagine all of it
Yeah.
by itself. Yeah.
In fact, if you go back a couple of slides,
The first slide had
I would say they're very verbose.
Yeah, you can't,
Right.
you can't use such
Too much
slides.
matter on it. As a PPT, try and not have so
Actually,
much.
it was a project and the slide, and I just struck that this morning that I want to use it in present because I was very passionate about it.
It's very good. And
So I
sure.
didn't do the modifications, I just kept it that way.
Exactly.
I knew that that was something I was going
That
to
would
become.
become. Some of the other things that I see you doing well, Parth, you're using your hand gestures very well.
Yeah.
Your hand, like even right now, you're holding it very, in a very open manner. And see, it's coming now subconsciously because you started using it. And it will keep coming, it will become very, very habitual if you keep doing it. So this is just 3 days in which you've done it.
Right.
Imagine if you do it 21 days, after that you don't need to be aware that you're holding hand. Your hand would automatically come. So very well done. Thank you very much, Parth. Well done. So the only thing is no footsie. No footsie playing. And again, for some of us, don't look back at the screen all the time because it gives the audience the impression the person doesn't know what's happening next. So the control of the presenter sometimes looks like he doesn't have control over the presenter. Alright? So be ready, know what's coming next, and go ahead with it. Keep the audience engaged through your eye contact. Don't break. Right? Well done, Parth. Well done. Thank you. Thank you. Alright, who do we